Biology 121 online video. This is our 11th video talking about protein synthesis, the second stage, translation. So let's review quickly the entire process of protein synthesis, also known as DNA expression, in which a DNA template results in the construction of a protein. We have two phases of this. It's called transcription. The first phase is called transcription. The second is called translation. So we start with DNA, which is inside the nucleus. Then the transcription phase produces a copy of the DNA in the form of messenger RNA. That RNA is produced and processed in the nucleus, and then it leaves to go find a ribosome outside the nucleus. That whole process was the subject of video 9, so if this doesn't sound familiar to you, maybe go back and check that one out again. Today we're going to be talking about the second step, translation. In translation, a polypeptide is formed from the mRNA template. Polypeptide is just a fancy name for an unfolded protein. This polypeptide gets produced at a ribosome and then gets folded into the appropriate protein shape with help from the rough endoplasmic reticulum. So let's look at this process of translation in detail. So once we've completed transcription, there's a mature messenger RNA molecule with its introns removed. The introns are the non-coding parts of the DNA, and only exons remaining. That mature RNA leaves the nucleus and goes and finds a ribosome to start its work. At a ribosome, that messenger RNA acts as a form of selection criteria for the building blocks of protein. Remember that proteins are polypeptides made up of different amino acids. So let's take a look at our friend the ribosome. This is an image of a ribosome with three colors, yellow, orange, and blue. Those three different colors represent different kinds of molecules. Ribosomes are composed of two subunits, a larger part that we see on top here and a smaller one that we see on the bottom. And each of those two subunits contains both multiple different molecules, some of which are proteins and some of which are RNA. These are the sites of protein synthesis, and the usual metaphor for what a ribosome is doing is that of an assembly line. So let's look at a historical picture of an assembly line, and here we have probably during World War II, because there's lots of women working at this factory, assembling something. And what's going to happen is as each person on this line finishes their work, they're going to hand the part off to the next person who's going to add another piece to it. Another way of visualizing that assembly line metaphor comes from is in this GIF, which comes from The Simpsons. And of course, these people who are feverishly assembling this incredibly long submarine sandwich, it's because Homer's sitting right there, just stuffing it into his mouth as they finish. Anyway, so this process, when we apply this assembly line metaphor to protein synthesis, is called translation. In modern living things, this specific step is called translation. So let's watch this other video here. What you're seeing at the bottom of your screen is a small ribosomal subunit, and this band here is a long string of messenger RNA. What's going to happen is the messenger RNA binds into the small subunit, a large subunit comes down. Now you see these blue pieces coming in, and notice this black tip that is growing progressively. And what that is each of these large blue, dark blue pieces that's coming in here, you see two of them inside now, another one comes in and one falls out. And each of these large blue pieces, dark blue pieces, is a molecule called transport RNA, tRNA. And at the very tip of each tRNA molecule, there's an amino acid. So each of those is bringing an amino acid into the ribosome, and then once there's enough of them, they start poking up out of the top of the ribosome. This ribosome has been pulled in and attached to a rough endoplasmic reticulum so that as the protein is being produced, it's already being folded. Remember that what makes the rough endoplasmic reticulum rough is that it has these ribosomal units attached to it, making it look rough when it was first seen under a microscope. This process of translation is still ongoing down here. You can see you still have these light blue helper proteins that are bringing these tRNA molecules in, and just see the very tip there. You can just see it at the very beginning. There's a piece on there, and then once it's gone, once it's moved along, it's missing. So let's look at this in a little more static of a way. Before we do, though, let's just establish all the different kinds of RNA that we've now dealt with. We have messenger RNA, which provides the selection criteria for amino acids. We have ribosomal RNA, which forms part of the ribosome. And we have transport RNA, tRNA, that binds with a specific amino acid and then delivers that protein to the ribosome. Remember that an amino acid is called a monomer, and when you put a bunch of them together, you get a polymer known as protein. So these are the 20 amino acids that exist in living things on Earth. There's about 500 amino acids naturally occurring on Earth, but only these 20 are used by living systems. This is part of the evidence for the notion that all life on Earth is related and shares a common origin. Now, here is a visualization of a tRNA molecule. You'll notice that it is composed of G's, A's, C's, and U's, because again, RNA is a nucleic acid with nitrogenous bases guanine, adenine, cytosine, and in the case of RNA, uracil. So we have these guanines binding with cytosines, we have adenines binding with uracils, and that's going to give a characteristically elongated shape to this tRNA molecule. Now there's this really interesting, funny thing that happens with tRNA. There's a variation in the genetic sequence right down here, kind of in the middle of the entire RNA molecule. And which 3 bases form this part of the molecule ends up determining which amino acid is going to be attracted to and fit at the other top end of the molecule. Seems a little crazy, but if we take a look at a more clearer visualization here, what we have is that same image that we just were seeing, but now we've color-coded the different pieces so that we can see how these different nitrogenous bases are binding together and forming bonds with each other. And you can imagine how a little bit of a shift in this end would open up or close down these spaces a little bit, and that could have a profound impact on where the binding sites are, such that at this very top end, the direction that it's curling outward and the specific bonds that are happening between these last few nitrogenous bases could have a pretty profound impact on which amino acid is able to find a space to bind in right over here. So a little bit of a change shaking down at this end of the wire can make a really profound difference in the shape up at this end of the wire, and just like everything else involving proteins, the shape is super important. So the shape of the tRNA molecule is so important that it's sensitive to a variation in the different nitrogenous bases all the way down at this end can affect which amino acid has room to fit on this end. So that leads us to this 3-letter scheme. So let me jump back a couple of squares. Remember that these 20 amino acids also have nicknames. So glycine will be denoted as GLY, and alanine will be ALA, and valine will be VAL. Histidine down here is typically HIS. So we have a shorthand to represent each of these 20 chemicals. We have this 3-base codon region that is the most important part of the tRNA. And now we have this chart that tells us if those 3 bases on the RNA are cytosine, guanine, and anything else, then that tRNA is going to be carrying an arginine amino acid. But on the other hand, if the first 2 are cytosines and the third one's anything else, then that's going to have room to fit a proline up at the other end. And in some cases, the third base makes a difference between one amino acid and another, like asparagine versus glutamine. There's also certain codes that are going to have a profound impact on what's happening with this molecule. So you've got uracil, adenine, followed by adenine, or by guanine is a stop codon. What that's going to do is terminate the process of protein synthesis. So at that point, that's where the molecule knows, the RNA is telling the ribosome, okay, we're done now, time to stop. And so you can imagine one small mutation from a piece of code, DNA code that was specifying taurine, to a piece of code that says stop, is going to destroy the protein entirely. Also, you may remember that we talked about the promoter regions being TA boxes. Well, here's our uracil adenine sequence. That's like a T and an A. There's also an A and a T that ends to a start. So, one last look at our protein synthesis. Down here, we have an image representing messenger RNA, and these four different colors are representing the four different nitrogenous bases: adenine, uracil, guanine, and cytosine. You'll notice they also are being consistent about their shapes, with a curved shape for the adenine and the uracil, and a pointy shape for the guanine and the cytosine, indicating those are the ones that are going to preferentially bond to each other. As we read this from left to right, this incoming RNA on this side is about to connect up, and you can see that it's a nice match. It's got a cytosine, guanine for this cytosine, an adenine for this uracil, and a cytosine for this guanine. Over here, we already have these nice bindings that have happened between a tRNA molecule carrying a particular amino acid, and as this ribosome moves forward along the mRNA, reading it through, the tRNA that has already donated its amino acid is being ejected from the ribosome. The one that's just arriving has not yet connected up its amino acid to the forming polypeptide chain. So we have here different polypeptides, right? Different amino acids forming together. So it's about to be 3 amino acids long, and then it'll be 4 amino acids long. And remember, a typical protein is going to be something in the order of hundreds or even thousands of amino acids long. So here we see the ribosome walking along this mRNA, tRNA coming in and bringing the amino acids to help form into this polypeptide chain. The metaphor I always use when I'm talking about how the coding region affects which amino acid is attached to a tRNA molecule is that the hat has to match the shoes. And if you put on different shoes, it's going to end up meaning that you'd better wear a different hat. So there's a kind of a silly, hopefully fun metaphor that'll help you keep in mind how the coding region at one end of a tRNA impacts which amino acid it brings, and then the tRNA has to match with the mRNA as a complementary matching in order to meet, in order for the specification, the instructions that were originally stored in the DNA to result in the correct sequence of amino acids in the protein. And that's why we call it translation, because we're no longer speaking in a language of amino acids with these 4 bases, A, G, C, and T. We're now speaking in the language of proteins with its 20 different amino acids.